I'm going to introduce Christian. Christian joined us last year in 2020 from the University of Southampton. So he joins Liverpool Head and Neck Centre in the University of Liverpool with honorary appointments to both Clatterbridge and the Liverpool Heart and Chest. He retained an adjunct professorship at La Jolla Immunology Institute in California. Now, Christian's German. He qualified in medicine in Munster and did his doctoral thesis in flow cytometry of breast cancer locally in North Germany, and then a postdoctoral fellowship at the Dana-Farber Cancer Institute in Boston, before returning to further clinical training and a further PhD in Southampton, and additional training and positions which eventually led to his chair in experimental cancer medicine in 2007. So Christian's interests, as I'm sure you know, are in the modulation of human adaptive immunity. And recent head and neck trials have been in novel immunotherapy drugs, in HPV-directed vaccines, and in mutinome vaccines, a very exciting concept that a vaccine is created that is bespoke to that patient's tumor. So having introduced Christian, I will hand over, and Christian is going to make us pay attention and let us know how to make T cells wake up to fight cancer. Thank you, Christian.
Richard, thank you so much. I blame Terry. I think that's our sort of new story, isn't it, Richard? That whenever something interesting happens, we blame Terry. So Terry was the reason I wound up in Liverpool after an almost total eternity in Southampton. And much of the work I'm going to present tonight is really about work that I did in Southampton. Although I hope that much of the delivery and the fun bit and the actually making it clinically useful will happen in Liverpool. And this is probably one of the more scary talks I've given, because not only haven't I the ability to interact with the audience, but I don't even really know who the audience is. So bear with me and jump in if I'm saying things that are incomprehensible, and interrupt as appropriate and as necessary. So the remit I'm going to talk to is how to make immunotherapy try and become a grown-up tool for patients with solid cancers. And I'm coming at this really as from the perspective of a physician. I grew up at the time when cancer immunotherapy really was something that could never possibly work. It was, clearly evident that immunotherapy was something for the initiated, because the immune system clearly had failed to do anything useful against the cancer, because obviously the cancer had developed and clearly the immune system couldn't do anything useful. And about 4 years ago now, I stood in front of a Cancer Research UK funding committee and tried to justify why I was still beating on this drum, and why it was useful to give me the money. Well, they didn't in the end. And really the best excuse I could bring up was the data that allogeneic transplantation, so transplantation people with hematological malignancies, was able to cure about 30 or 40% of patients with hematological cancers, so acute leukemias. But the world has really changed since then, and it's a time of really quite unprecedented change in cancer biology. And immunotherapy with drugs that modulate this pathway, PD-1, anti-PD-1, have really altered the way we think about what we can achieve. And immunotherapy has not only become the standard of treatment in many solid tumors, but also generates clinical benefit that is quite stunning for a physician and, of course, for the patients. So the reference standard has become the use of drugs like anti-PD-1, nivolumab, pembrolizumab, or anti-PD ligand 1, and then there's a whole family of these drugs that do much the same thing. What we've learned is that across the board, about a quarter of the patients benefit. And if you combine drugs either with multiple agents of the immunotherapy flavor or even with chemotherapy in lung cancer, you manage to get a response rate about 50%. And that's what I'm trying to say here. And I've picked up melanoma because that's where we all started, but it's in bladder cancer, lung cancer, and of course head and neck cancer, where we're still a little way off the 50% range. And for a clinician, this is quite an amazing experience. And this is one of my own patients with metastatic lung cancer. So here's a small, tiny little cancer that started in the right upper lobe. This is a CT scan which which has been overlaid by a PET imaging, which picks up cancer cells because they take up sugar and then they glow this white color. And this gentleman had, here's the primary lymph node involvement in the mediastinum, a liver stuffed full of cancer, and presented with a pathological fracture of his right femur. So under all clinical context, this would have been a death sentence. And yet here is 2021, 5 years on after immunotherapy, disease-free and with no evidence of recurrence in the interim. So I think what is really changing the way I think about cancer treatment and cancer immunotherapy, of course, is that this does something really very special. It reprograms the patient's immunotherapy, in the patient's immune system, but sometimes. And what we're learning is that immunology and the human immune system is really an ongoing balance between immune suppression and immune control. And if immune suppression wins, then the cancer progresses and the patient dies. And if immune control wins, then we manage to achieve control of the cancer, remove the cancer cells, and then these patients can be cured. And we now know that, for example, in melanoma, if you hit a disease control rate of about 3 years, then you probably aren't going to relapse again, even if you presented with metastatic cancer. So the real puzzle has been, what is the underpinning biology that makes this kind of event happen? And here's some data of our own. This was data generated in Southampton, led by the group of Professor Gareth Thomas, a professor of histopathology in Southampton. And this represents the information about 550 patients with head and neck cancer. And I've picked out head and neck cancer because that's what I'm interested in, having come to Liverpool. But much of this data could be generated in many other solid tumours. Head and neck cancer falls into pots, and it's important that you remember that for the talk going forward. About 1/3 to 40% of the tumours are caused by a virus that in most of us is relatively harmless. So we have the human papillomavirus infection and then get rid of it and clear it. But in a small percentage of people, the virus becomes nested in the mucosal tissue of our oropharynx and then causes malignancies. And that is in contrast to the majority of patients who have cancer that is driven by smoking and high concentration alcohol. And it turns out that in both of these tumors, we use very similar treatments. We cut off the cancer, we give chemotherapy, and we give radiotherapy. And then we wonder why sometimes it works and oftentimes it doesn't. So what we're learning here is that the number of immune cells that are sitting in this cancer trying to control this has a major role to play. So if you have got no or few T cells, then the patients have a really bad outlook. So 60% of patients die in the first sort of 15 years. On the other hand, if you have a virus that's causing the cancer, and that is something that the immune system is quite good at seeing, and you have lots of immune cells as a result, then the chances of survival stage by stage are really much, much better. And you can see that only about 10 or 15% of patients die. So there is a fundamental difference in cancers that are involved and invaded by immune cells compared to those that haven't. And if you look at this under the microscope, then this is what it might look like. This is an adenocarcinoma, adeno because it looks like a gland structure. These beautiful little round nuclei are the nuclei of the cells that are the carcinoma cells that are arranging themselves in these glands. Here's the hollow space in the middle. And in between these nests of tumor cells, you can see these dark purple cells. And they are the immune cells that are trying to access the tumor cells and hopefully trying to eliminate that. And this is the sort of morphological or histological assessment of what a cancer with some immune cells might look like. Now, if you unleash these beasts, then this is what you get. So this is a tumor where the tissue has been overrun by cytotoxic T cells, CD3 positive T cells, and you can see that here where it's all very distinctly separated, these are sort of filling the space in and among the tumor cells. And this would be the sort of picture that you would expect a patient to have that is eventually cured. The problem remains that separating these two is visually quite easy at this, but sort of grading this is a real challenge, and I hope that in Liverpool we'll make some inroads onto in developing these kind of counting of these immune cells into clinical practice. But the other bit is that we don't really know what drives these T cells and on what turns them on. I'm sorry, apologies for the technical challenge, and what they might be actually trying to recognize in these cancer cells. And so if you then think about it, the cancer cells, of course, arise from healthy cells, and they're different to the healthy cells because they misbehave, but much of what is in the cancer cell is still similar to what is in a healthy cell. And so for our immune system, the real challenge is to figure out what the cancer cells are doing differently to the healthy cells, picking up that difference, and then doing something about it in terms of activating the immune cells to allow the cancer targeting T cells to eliminate the tumors. We've learned that there is and I've picked out head and neck cancer again for the same reason, that there are whole different categories of what cancer immune cells might see. So for human papillomavirus driven cancers, the viral antigens are relatively interesting to the immune system because our immune system is great at picking up viruses. That's what it does for a living, and that's what it should be doing. So these are perhaps the lowest hanging fruit. So virally driven tumors, particularly driven by human papillomavirus, is a story or offer a story that is worthwhile following. But then the majority of tumors, not just in head and neck cancer, but generically, are not driven by viruses. And so the question then arises, well, what else might we be able to see to target in those tumors? And one of the bits that I'll talk a little bit about are the very features that turn a tumor cell into a cancer cell. So when our cells are born, they generate or they inherit the genetic information from their parent cell. So each cell division is ideally a faithful copy of what the parent cell contained. If that information is faulty and gets passed on, we call that a mutation. And these mutations can alter the behavior of the cell in which this mutation has happened. Now, if the mutation is catastrophic, the cell dies. That's bad for the cell, but good for the patient, because nothing really untoward happens other than you've made a cell that's eventually keeled over and died. But if these cells can continue to grow because they've, for example, forgotten how to turn themselves off in the right cues, then you get cancer cells that have mutations, and these mutations in their own right would then be propagated in the cancers, and these are what one could now begin to think of as targets for immunotherapy, and I'll come back to that a little bit later. But because the cancer cells are very similar to healthy cells, there are also molecules that are found in healthy cells that are called tumor-associated antigens. So things that make the cancer cells look a little bit different rather than radically different as the mutations. And again, these are being targeted and potentially could be targeted by making the immune system work better, so by vaccination. And I'll go through some of the data in the talk, and I've picked out and read the bits that we are actually interested in clinically. So PSMA, CEA, HDERD, MAGE, and so on are molecules that we tested as targets in the clinic. At the moment, these are all sort of sitting next to each other without a great ability to choose which ones of these are good. And this is a cartoon I got from one of my colleagues who was developing mutation-specific vaccines. And we think that the most difficult bit to wake the immune system is something that is also found in all our healthy cells. That the reason for that is that our immune system goes to, or these cells in our immune system go to school in an organ in the middle of the chest called the thymus. And they're taught what is self, so what the T cells need to ignore, and what is not self. This distinction is really important because our immune system is no good at picking up things that are self because they've gone to school and they've been trained to ignore that. Whereas the antigens that are found as a result of the mutation, or as, for example, viral infections, have not undergone this particular selection process and therefore have perhaps a better chance for our immune cells to activate against and to recognize that foreign. And that's the reason why this cartoon identifies specificity on the one hand, so the mutations are unique to each cancer, and sensitivity or immunogenicity on the other, where we would hope that these would be the most recognized. However, at the moment, this is all speculation. So we have no idea whether these self-antigens that are mutated or viral antigens or indeed share antigens are any better or any different in terms of immunogenicity. And so one of the questions that we would really like to develop in our program in Liverpool is to ask which of these are the best and to systematically compare these. So working out what works needs a tool set, and what we really do in my practice, and again the reason for coming to Liverpool, is to test this in people, because you can do all manners of things in preclinical models, particularly in mice, but in the end that isn't really what you're after. You're trying to work out what makes human cancer tick and how we could make human cancer better. And so the tool set that we're developing is to take samples before you intervene or vaccinate, and then to study the hell out of it. So we're looking at the immune cells, looking at the genes in the cancer cells, looking at the associated other structures, for example the blood that might be accompanying this tissue. That you would then do your intervention, immunotherapy in the broader sense, vaccination in the specific sense, and then take another sample from the cancer tissue to work out whether what you thought would happen has happened. And then hopefully, if it has, we'll clap your hands and move on, or if it hasn't happened, try and figure out why it hasn't worked. And this is ultimately the underpinning biology that we would like to study and use the best possible tools to evaluate. Now, again, we've done quite a lot of this, and really I started this as a PhD student and then as a young doctor when I was first in Southampton, and we were really completely naive in human immunotherapy at least. But the work that came out of the lab that I was privileged to do my PhD in was run by by a scientist called Frida Stevenson, who had the idea that if you were to make something that is interesting, so for example something that is from tetanus, something we've all been trained again and vaccinated against, and hook that up to the bit that is from the cancer gene sequence that our immune system might not be so interested in, would that be a trick to make these two look overall appealing? And that turned out to be a really smart idea, and we're still following that concept now. And so, because we were working with vaccines, we were looking at DNA as a strategy for delivery, and what we did in the lab, and actually that was one of my tasks in my own PhD, we assembled these DNA plasmid vaccines, where we took a chunk of the cancer bit and a chunk of the tetanus, so this domain sequence, and strung them together and tried to work out whether that might cause the immune system to wake up. And this can be given into the skin or into the muscle, and now we're working on thinking about ways to deliver this by inhaled strategies, and then the cells that you are injecting this stuff in, either the skin cells or the muscle cells, turn the DNA into the protein that ultimately turns on the immune cells. So it's a little bit like the Pfizer vaccine that we're using for coronavirus, only the template is RNA in the Pfizer vaccine, and this is DNA. And we were quite excited when we did this work, and this was just at the beginning of my time in the lab, because Jason Rice, who has since left science, has turned into a full-time gardener, showed that this was able to confer protection in a colorectal cancer model. We were able to do this in a hematological cell model, and the immune cells were able to remember that they'd been exposed to the cancer before. We were able to overcome this model of tolerance, so this training against being activated in a mouse model again, and in the end took this into a number of clinical trials, which I'll talk about briefly, both in carcinoembryonic antigens, so in solid tumors of the lung cancer and bowel cancer ilk, as well as in prostate cancer. And what we learned was that we were incredibly naive. So we gave it into the muscle, and we could indeed vaccinate the patients, and that was all quite safe, and we didn't make anyone ill. And actually we did wake up the immune system, so we could measure immune responses to tetanus in 70 out of the 30 prostate cancer responders. And we were able to measure T-cell responses, so the kinds of T-cells that we were trying to induce in roughly 60% of the patients also, and T-cells against the DOM sequence in 30% in 30 out of 31 responders. So by and large, this looked as as though we had been able to stimulate the immune system in just the right way. And when we looked at clinical outcomes, there was a bit of a suggestion that this was actually useful to the patients. But we certainly didn't cure any cancer. And undeterred by our apparent incomplete success, or rather than calling it failure, we went on to test this same kind of concept. So again, using the tetanus sequence, and this time using something that is expressed in colorectal cancers, in lung cancers. So this is a molecule called carcinoembryonic antigen, and there is a little chunk of it that is widely expressed in these solid tumors. And we vaccinated patients who were either disease-free, who were in the adjuvant setting, so who were just at high risk of developing further disease. And we compared those to patients who had measurable cancer, people who had had all the best kind of surgery and radiotherapy and resection of cancers and any chemotherapy that we had available at the time. And it was really quite intriguing what we learned here. So we were again able to stimulate the immune system against the tetanus vaccine. Mind you, this is sort of several million quid worth of clinical trial for something that you could buy in Boots Pharmacy as a tetanus jab for £1.50. So it's clearly not a particularly smart way of vaccinating against tetanus, although we no doubt did that also. But what we found was that in half of the patients who had no measurable disease, we were able to induce immune responses against this tumor antigen. And the vast majority of people who had been given the tetanus vaccine were able to recognize that. So these patients were clearly able to do something with our vaccine. The vaccine actually worked. When we compared that to people who had metastatic disease, so recurrent disease, you can see that the frequency dropped by about 50%. So 92% out of the disease-free patients were able to make an immune response of 14 out of 15, and in roughly half of the patients with metastatic disease. And when we looked at the bits against the cancer antigen we were actually interested in, then that dropped by about 50%. What was really quite intriguing, though, was that we also saw some unexpected side effects. And the molecule that we were vaccinating against, the CEA molecule, or carcinoembryonic antigen, turned out to also be quite highly expressed and present in normal bowel cells. And so we were perhaps naive not to have expected that. But what we did see is that some of the patients we vaccinated developed signs of a disturbed bowel, so they developed diarrhea. And we therefore thought, well, this is actually quite neat, because clearly this suggests to us that the T cells are actually going where the antigen is. And what was the real surprise is that if you did achieve that and you develop this immune-related side effect, that the patients did much, much better. And, you know, for a group of people who were at the end of their treatment pathway, tripling the outcome from 270 days in those who didn't to just under 1,000 days. So that's just over 2.5 years of median survival. That's quite a story. However, we weren't in a great position from an intellectual property perspective, and Cancer Research UK, who initially gave us the okay to do a randomized study, pulled out at the 11th hour, so we never went on to test this particular vaccine any further. And it's on my wish list of things to do if I ever have the time and the money to do that. We did learn some interesting extra things, and what we were able to do is pick out the cells that were reactive to this particular chunk of carcinoembryonic antigen. So this is a small sequence of 9 amino acids, and you can make that into a chemical reagent and work out which T cells in the blood recognize that. So that's what I'm displaying here. So we were able to pick out that using a particular tool that would pick out a substantial fraction of the peripheral blood T cells that were of the right ilk were recognizing this peptide. And what we were then able to do is pick up the cells and sort of do a molecular fingerprint of those. And for T cells, the molecular fingerprint that you can use is sequencing the T cell receptors, so the sort of link that recognizes the antigen, and that gives you a gene sequence that you can use to track this particular T cell. And we were able to demonstrate that unexpectedly, this T cell already existed before we ever had started vaccinating. So this patient had her lung cancer receptor in 2007, we vaccinated in 2008, and the T cells that we were expanding were clearly already present several years before we actually ever gave them the vaccine. So there's quite a lot of learning that's still relevant that come out of this. And so the first one, I think, is that shared antigens can be targeted successfully. Vaccination improves outcome a bit in some patients. Vaccination can expand pre-existing immune cells, and these appear to live in the cancer, and we can pick them up in a cancer. So that's really important because it suggests that much of the immune memory in our body lives, in this particular case, in the tissue or in the cancer tissue if you're a cancer sufferer. Side effects link to the targets in healthy cells, and if therefore you vaccinate, you need to be careful to target things that aren't found in healthy cells, and that's one of the lessons we're taking forward. And finally, the less cancer the patient has, the less volume of cancer, the better the immune responses. And admittedly, our data are sort of binary, lots of cancer, almost very little, but I think it is quite intuitive that this is probably generic, and the lesson to learn is to vaccinate as early as possible. We've also learned that all the biological events are not taking place in the blood, but that we need to focus on evaluating the cancer tissue. And in the end, that we need to strictly standardize the immune monitoring, so do the same evaluation over and over again. There's no particular learning outcome here. We got that right from the beginning. So if we now move on from what we've done to what we would like to do, then I think the first project really is to evaluate whether of these shared antigens we can look at the ones that are unique and driven by viruses. So in head and neck cancer, that's the human papillomavirus, and there is already some evidence in the literature that if you make, in this case, a DNA vaccine against human papillomavirus, you can turn a tumor with few brown cells or few T cells into a tumor with lots of brown cells or lots of T cells, so turning an immune cold into an immune hot tumor. So that's, I think, the first hurdle that we need to achieve, and we'll see whether our own trials can achieve that. And we're in a phase 1/2 study that is about to open in Liverpool also, where we've completed the vaccination of people who are currently disease-free, and all of those we've tested have made an immune response to our RNA vaccine, and we're now collecting the group of patients that have recurrent disease to see whether this can actually do something useful. So you can see that the lessons that we learned, better immune responses without cancer, less good immune responses perhaps in people with advanced cancer, have really fed into the design of our study. We've also begun exploring whether the cancer mutations can be explored as the target. And here, this is probably one of the most widely presented talks about images about genomics that came out of a Nature Cancer now 7 years ago. And what this group did is sequence or present all the data that were available on the number of abnormal genes in cancer. So here they've aligned the types of cancer: melanoma, lung squamous carcinoma, lung adenocarcinoma, bladder cancer. Here's head and neck cancer. And then they've looked at many patients, so hundreds of patients, and totted up how many mutations there were. And the numbers are related to how many normal genes there are, or how many megabases of normal genes there are. And what you can see, and this is a logarithmic scale, is that the incidence of mutations goes from really quite low, 0.1 per million bases, to quite a lot, 10 per million bases, and there is an enormous range. And what we've learned is that this frequency of mutations seems to link also with the immune competence or with the ability of the patient's immune system to recognize them. And I think that's probably simply a chance of, you've got lots of mutations, there is a better chance the immune system might see that. And that's possibly while melanoma and lung cancer have sat at the leading edge of cancer immunotherapy, or where other tumors, so for example brain tumors, are still lagging way behind. And head and neck cancer is sort of in the not too bad upper third perhaps of this setting. So when we now think about these mutated antigens, the difficulty had in the past been that you had no clue what they were in an individual patient. And here a huge technological development has come to our aid. And that is, you can now take a cancer sample and just sequence it. Something that for the first time done in human beings took 10 years and many hundreds of millions of pounds can now be done on the benchtop in a kit that's maybe the size of a big computer screen, and can be done in an afternoon. So you can now take a sample, both of the cancer cells and of the healthy cells, and work out what all the genes look like, and do that reliably, reproducibly, and at relatively low cost. So cost for that is about 500 quid now. So this now gives us the possibility to take a cancer and read out all the abnormal genes that are present in this cancer tissue. And if you now do that systematically, and so you read the cells that are in the normal, the genes that are in the normal cells, the genes that are in the cancer cells, and then you subtract one from the other, that leaves you with the genes that are abnormal in the tumor cells, and you work out the ones that the cells are actually working with, then you could begin making a string of bead of the bits that are abnormal, and then see if you can package that into a vaccine. And indeed, what would have seemed like science fiction when I was doing my own PhD is now becoming possible and clinically feasible, and you can deliver such a vaccine in roughly 4 to 6 weeks now. And so what we've done a little bit of preclinical work in this space. So we had a patient with lung cancer, and for some reason I got involved in this gentleman's care, and we were super keen to try and make him a personalized vaccine. So we went all out, we sequenced his white blood cells, we sequenced his tumor, and in the end were able to identify all the genes that were abnormal. We found a pharma partner who would make us a vaccine. And then by the time that has all happened, unfortunately, we were too slow, and the patient had passed away. It took us a year to get from this sample to the final result. But what we did learn from this is that when we use this kind of vaccine that we've now assembled and tested it in a mouse model that had, by fortuitous circumstances, a similar immune system to the patient, we were able to pick out immune cells that were actually reactive against the mutations, and we could demonstrate that this vaccine was actually sufficiently immunogenic and sufficiently good, therefore, at waking up immune cells, that this was a worthwhile effort. And this, in turn, this one person, this one individual, even though he personally didn't benefit, has led to a whole program of work. And the company Transgene has built a large facility which is now making vaccines for a number of clinical trials, and will in Liverpool will be one of the first groups to take this into head and neck cancer, making a personalized cancer vaccine for our patients. So we hope that this will start in the next month or so, and again this will be driven by the information that we had before and the concept that less cancer is better. So we will hopefully ask our patients who are undergoing surgery to allow us access to some of their tissue. We'll go away and do the sequencing and the assembly of the vaccine with our transgene friends, and then in the meantime they'll have their standard of care treatment, and then we'll be able to offer vaccination to these individuals. And because of course these are cancer free, we won't be able to work out whether we can do any more than stimulate immune responses. So we've done a trick, and we've said we're going to vaccinate half of the patients and follow half of the patients and see whether the vaccination makes a difference. And of course we'll have a vaccine for everyone. So should the patients be unlucky enough to have relapse, we can offer them the vaccine that we've already made. So sort of giving us 2 bites at the cherry, and hopefully working out whether this kind of approach is clinically useful. This is clearly still a lot of work, and even though I said you can do this in an afternoon and on a benchtop, nonetheless it would be so much easier if you could just make one vaccine that is useful for everybody. And that's another parallel approach, again, that we're hoping to take into the clinic towards the end of this year in Liverpool. And we found some molecules that are really uniquely upregulated in cancer cells, again, head and neck cancer. So these are cells here that we've stained with an antibody against this antigen, these 2 antigens. Here's our oropharyngeal squamous cell carcinoma 1, 2, and 3, and the brown stain picks out the molecule that we're interested in. And these molecules are also found in the ovaries and the testes, so these are called cancer testis antigens, and that's a whole big family of molecules that one can test by immunotherapy. And there's an august and long-standing history demonstrating that these might be good targets. So we've been quite lucky to find our own, and we're now going to take this into the clinic, and we've been able to stimulate immune cells against these cancer testis antigens, and we can, when we look in the blood and in the cancer tissue from patients with cancer, see that the immune cells are already present, sort of reminiscent of what we found in the CEA vaccine trial in our lung cancer patient, where I showed you some data. The reason why that's important is if these immune cells exist and vaccination can be used to activate them, then I think that's a much easier chance than if you have to stimulate a whole bunch of new cells. So turning on what's already there, I think is going to be a lot easier than generating a whole new set of immune cells. So I'm quite excited about this, and we'll see whether this will actually turn into clinical usefulness. And this is the equivalent preclinical model. So we have a mouse model in which tumors grow spontaneously, and we can vaccinate these mice against the targets. And here again, much like the data I showed from the HPV, we can increase the number of brown cells or T cells that are in the tumor compared to animals that are not vaccinated. And if the tumor grows unchecked in this way and you vaccinate, then you protect the mice against the tumor. We're quite lucky because we've found another set of friends who would like to play with us, and this is a company called Touchlight. It's a small UK-based startup company who initially made their living out of making DNA vaccines for a whole variety of other companies rather than as a vaccine company. And again, we were quite successful into talking them into that they should begin thinking of themselves as a vaccine company. And so that's been quite successful, and we persuaded Cancer Research UK to help us in this venture. So we've now got this sort of chemical method to make lots of vaccines really quickly, and that's what we will take into the clinic with the Touchlight team. And because this is, I don't know if you can see the similarity, it's got a sort of closed-end loop of this double-stranded DNA, and the first person who drew this thought, oh, this looks like a bone, and the name stuck, and so these are called doggy bone DNAs. And these are really easy to make, and they are chemically really stable, so you could make, for example, a COVID vaccine and ship it to Malaysia, or make it indeed an anti-cancer vaccine and ship it, because they're very temperature stable and they're very easy and quick to make. So we're super excited about taking this into the clinic as a technology. And along the same lines that I've already identified, we'll go into the clinic, we'll vaccinate, then in this particular case we'll add normal standard immunotherapy, and we'll see in the tissue whether it turns on the right kind of immune cells. And we were super lucky again because CRUK, Cancer Research UK, supported that if this first bit was successful, they would also allow us to do an efficacy element to this study, to really work out whether the vaccine, in addition to standard immunotherapy, would actually add something else to the process. So I've talked a lot about antigens that are found in cancer cells and that we might target, but this is very much like this sort of yin and yang in life overall. And where there are immune cells that are trying, here are the little brown spots, or perhaps blue spots sitting in the tumor microenvironment, there are other cells that are trying to impair and to protect the cancer cells. So this is a head and neck cancer. These pink cells are the cancer cells, and they're sitting, I don't know if you can see, they're sitting in this nest that they've made for themselves. And this nest consists of tumor-associated fibroblasts, or cancer-associated fibroblasts. The technical shorthand are CAFs, cancer-associated fibroblasts. And these are really intriguing because they're not cancerous. They're just turned on and activated by the cancer cells. So the cancer releases stuff that makes these guys do their job. But they, in turn, and I imagine this a little bit like a thorny hedge that they're building around them, they prevent the T cells from going into attack the cancer cells. So what is happening is that in the tumor microenvironment, the tumor cells are training normal cells to do their bidding, and the normal cells, the cancer-associated fibroblasts, prevent the immune cells from eliminating the cancer cells in their own right. So this is really quite smart from the tumors, but difficult from the immune therapy perspective, because however many immune cells you make, if they can't get to the cancer cells, then you're in trouble. And it turns out that this is again related to outcomes. So if you have few of these stromal cells, these CUFs, then the patients by and large do better. So in this case, 70% of patients are alive at 10 years. And if you have lots of these cancer-associated fibroblasts, then only about 20 to 30% are alive at 10 years. And this is true not just for head and neck cancer, but also for lung cancer and for esophageal cancer and for pancreatic cancer and so on. So this concept that we are seeing here is graphed here. So here are our red cancer-associated fibroblasts trained by the tumor cells to protect the tumor from the immune cells. And so what we would like to do is find ways in which you can turn these cells off and allow the interface between the tumor and the cancer cells, the T cells and the cancer cells to happen, and then hopefully this will lead to clinical usefulness. And if you do this, and there's a compound that turns cancer-associated fibroblasts off, made by a company called Genkyotex, which is why this GKT is the shorthand, then you can see that where in animals in which the cancer cells are allowed to train the cancer-associated fibroblasts, the tumor cells grow and grow really quite rapidly. But if you block the cancer-associated fibroblasts, you turn off the CUFs and you allow the immune system of the mice to clear the tumor. And so this is a sort of really interesting way of thinking about not just vaccinating against the tumor cells, but also engaging immune escape pathways and overcoming those. And for some exciting reason, the molecule that I talked to you about earlier, so this antigen 1, is also expressed on the cancer-associated fibroblasts. And we sort of just found that out almost fortuitously later on. So in our trial, when we vaccinate against the cancer antigens, we may also be vaccinating against CUFs at the same time. And so that will be really quite exciting to work out. So then the strategy here is training the immune system by systematic evaluation of classes of antigens, viral antigens, mutational antigens, and shared antigens, but also antigens that may be expressed on other cells that are not cancerous, that are just helping the cancer cells survive. We'll do quite a careful analysis of various platforms, what might be the best way of getting to Rome, I guess is the summary here. And again, what we've already done, strictly standardizing immune monitoring tools. And we've gotten a lot better at this and have new friends that have allowed us to do this in a much more sophisticated way than I was able to do when we first wrote the first trial. And so that sort of takes me to the end of the story here, and it's very clear that this is all a collaborative effort. And while I'm allowed to talk about it, there are many people who are critical for this. Terry, no doubt, is again to blame because he made me come to Liverpool in the first place. But much of this work relies on clinical colleagues and surgical colleagues to enable us to access some of the tissue, and hopefully then we'll take this into the clinic. And Joe, I hope, will be a partner in this. The lab team with whom we've developed much of this science in human immunology. Gareth Thomas is the pathologist that I've mentioned a number of times, and here's the surgical and oncology team from Southampton. An amazing collaboration has happened with this particular chap, Vijaynan Pandurangan. So he's a respiratory physician turned scientist who has opened up the doors for us to access some of the astounding technology that his lab has developed and that we're now levying at this problem. And his partner is Ferret A, who's a bioinformatician also in the La Jolla Institute. And I don't really feel I can go away without mentioning this team. So Kui Ping Lem and Sokching Chong, who are in Malaysia. So they're not in Kuala Lumpur, but their city is just next door to Kuala Lumpur. And they work for Cancer Research Malaysia. So they were together with Natalia and with Gareth the people who identified these 2 antigens that we want to test take into the clinical trial. And we hope that what we will do is, once we've got the first steps done in the UK, that we'll then hopefully do the phase 2 of this trial with the Malaysian team. And then I think that will be super exciting if we're actually able to take something from a developed country into a place where the delivery is much more complicated, using technology that I think will easily translate. And in the end, we have to thank many of the people who've given us the money. So Cancer Research UK, our ECMCs, the MRC was really critical for the cancer story that Gareth, Natalia and I developed with the Kuala Lumpur team. Our local charity, in particular a lady called Mrs. Margaret Whittaker, who's given us enormous support to develop some of the science, and many of the participants and other centres and investigators that we're working with. So that's where I would like to leave it, and thank you for your attention, and take any questions.
Thank you very much, Christian, and thanks for getting the lecture series off to such a strong start. I was going to ask you about any friction in the system about giving cancer patients RNA and DNA vaccines, and then maybe a subsidiary question: to what extent the Pfizer COVID vaccine might free that up. And I'm thinking, you know, sometimes that wars are very dark things, but wars can move surgery on a long way. People learn things during the war. They're forced to do things that they wouldn't have done as fast. And whether that field of RNA vaccines just gets turbocharged because of what's happening at the moment.
So it's very intriguing that the same platform that we were testing cancer is now protecting us against this virus. And I think the perception that vaccines are a good thing is getting augmented. I think as a cancer vaccine field will benefit from what is coming to protect us against coronavirus, on the assumption that it actually will. And you know, if it turns out that vaccines allow us to get back to everyday life, I think that will be a huge boon for the testing in cancer settings, and perhaps even in the pre-malignant setting, because I think there are many stories to be developed there. We've been very lucky that our patients, by and large, have thought vaccination was a cool thing to be part of, and have been extremely generous in allowing us to take blood and to take samples and to be part of our trials. And I think that the coronavirus vaccines will just help that along. What we will learn, I think, is more about the functioning of vaccines, and you know, how well does a vaccine induce T cell? How does it induce antibodies? Are these useful? Where do the antibody cells need to live, and where do the T cells need to go? These things will be no doubt informed by some of the efforts that are going into coronavirus. So I think we're going to learn much more about human immunology than we would have otherwise done. So I think it will be mutually informative.
So a question that's come in, Christian, is how realistically do we expect to expand T cells of our patients, and from which compartment, and how would we overcome the immunosuppressive effects of surgery, neck dissection, cytotoxic chemotherapy, blood transfusion, things we have to do to our patients at the moment, which seem to be immunosuppressive?
Yes. I think you start with one element and dissect what that achieves. So give a vaccine, see what happens, and then learn from what hasn't worked. So look in the tumor, what are the mechanisms for immune escape? So our early data suggested that even in advanced cancer, you can do things that alter outcomes. So a simple DNA vaccine making people with colorectal and lung cancer live longer that's quite a big achievement, I think. So if we could deliver that kind of progress in a steady and careful evaluation, so that we can work out, well, what do the cancer-associated fibroblasts do? What do immune inhibitory molecules do? What other cells are there that we need to into account? Then we can begin developing a picture of what the overall problems are. And then I think we can also begin to understand what happens to an individual person that we need to consider in our making our choices. And so I think the development will be in three different ways. One will be technological. What can we do? How do we make vaccines? What are the antigens? The second is, how do we measure the effect and its failure? And the third is, how do we understand the soil in which all the immune responses need to develop. And of course, if someone's immune system is irreparably damaged, then immunotherapy won't work. And no doubt there'll be a group of patients in whom we can never achieve that. But I think the excitement is that if you push the right buttons, you can clear cancer metastatic disease even in people who have been pre-treated in, say, 50% of melanomas. That sort of opens the door to an enthusiasm that is truly astounding.
Yeah, and a question as well that's come in. If a mutinome vaccine is given and the antigens to which it's directed are different in every patient, is it that nobody gets adverse effects that are related to those antigens, or is it that every patient gets a different adverse effect profile, which could be a little bit hairy?
So far, the mutinome vaccines have seemed to be very safe and seem to not have generated new and unexpected immune toxicities. And there is a moderate data set of several hundred patients in the literature now. I think the reason is that the mutations have already been seen or been available to the immune system. So you're making better what is perhaps already there, rather than causing immune toxicity against something that the immune system shouldn't normally recognize. So that's the sort of self that we would normally train our immune system to ignore. There is little suggestion that immunotherapy generates more than a sort of known spectrum of immune toxicities. So, the most common ones are skin and bowel and liver, and then there are rarer ones, but there seems to be an exhaustible spectrum. So my expectation is that we won't discover new immune toxicities with mutinome vaccines, and that because the targets are already widely present in the patient, that it is unlikely that we would cause new immune toxicity. That seems to be borne out by the observations we have so far, and even if you combine these with checkpoint inhibitors to sort of protect and supercharge the T cells, that's the immune toxicity you see seem to be checkpoint inhibitor toxicities rather than vaccine specific toxicity. So I expect that may still happen, but I think it's going to be uncommon.
Thanks very much, Christian.